This presentation is in response to exam 2B, question number 1 concerning mechanisms for generating antibody diversity. So, antibodies exhibit a great deal of idiotypic variation. These idiotypes are the antigen binding specificities of the antibody molecule. Idiotypes represent these unique amino acid sequences in the variable region that are associated with the antigen binding capability of the antibody molecule. Variability in the amino acid sequence in these regions is concentrated to 3 or 4 hypervariable regions, known as complementary determining regions, surrounded by relatively invariant residues, these framework residues. And so these complementary determining regions are the areas that make contact with the antigen. So given all this variation, we need to know about the genetics involved. The human immune system is capable of producing a vast number of different antibody molecules, each with its own antigenic specificity. This vast diversity is possible because the genes involved undergo an unusual type of interaction. Embryonic DNA contains a large number of genes for the variable regions of the heavy and light chains of the immunoglobulin molecules. A process called a somatic recombination, followed by RNA splicing, results in a large variety of B cell lines that encode different heavy chains and light chains. There's also a fairly high rate of somatic mutation in the kappa, lambda, and heavy chains, and this further contributes to the diversity of antibody molecule structures. So heavy chains and the kappa and lambda chains are each encoded on separate chromosomes. H chain being coded on chromosome 14, lambda chain is on chromosome 22, and the gene complex for the kappa chain is on chromosome number 2. The heavy and light chains vary quite a bit in their variable region amino acid composition, but they're relatively constant in the constant region, the carboxy terminal portion of the molecule. Analysis of immunoglobulin genes has revealed that variable and constant regions are separately encoded and located on different fragments of the DNA. As in other genes, coding sequence known as exons in the DNA code for the amino acid sequences in antibody molecules. The exons are separated by these intervening non-coding nucleotide sequences known as introns. Both the exons and introns are transcribed into RNA, but RNA splicing removes the introns and it leaves the exons joined together. The exons that code for variable domains are split up into smaller segments of DNA along the chromosome. Making proper exons from these segments requires rearranging and rejoining the segments to form the gene sequences for immunoglobulin. There are 3 chains that code for each of the immunoglobulin light chain. 2 gene segments encode the variable domain of the human kappa chain. The initial segment, the kappa variable region, encodes the first 95 amino acids. A second gene, the joining kappa segment, encodes the remaining 13 amino acids of the variable exon. And a third gene dictates the amino acid sequence of the constant region. The lambda light chains arise from a similar complex on chromosome 22. Although similar to light chain genes, heavy chain gene organization is a little more complex. Whereas a light chain gene region encodes only one to a few constant domain, the heavy chain gene region must code for each of the different antibody classes, and for a few subclasses as well. Furthermore, the exon coding for the variable portion of the H gene is composed of 3 segments, not just 2. So the additional variable region DNA segment is designated the diversity gene region. The diversity gene region accounts for the 3rd complementary diversity region of the H chain. It comprises only 2 or 3 amino acids. The additional variable region DNA segment is designated the diversity gene region. And the diversity region accounts for the 3rd complementary determining region of the heavy chain. It comprises only 2 or 3 amino acids. Gene rearrangements will link to diversity and joining segments, and then these join to a variable segment to produce the VDJ exon. There are over 200 variable heavy genes, at least 20 diversity heavy genes, and 6 joining heavy genes. And given this enormous gene pool with its large potential combinations, we start to see how this great diversity, especially for the heavy chain variable region, is possible. So each gene segment, whether for the heavy lambda or kappa chain, is flanked by non-coding recombinational signal sequences, or RSSs, that specify the direction of the joining segments. So each RSS consists of a very conserved sequence containing 3 parts: a heptamer, a spacer, and a nonamer. The spacer will contain either 12 nucleotides equaling 1 turn of the DNA helix, or 23 nucleotides, 2 turns. Joining of the gene segments occurs only when the number of spacer nucleotides are different lengths. Joining the L chain genes, for the kappa chain, the RSS on the downstream side of the variable segment has a 12 nucleotide spacer, while the signal on the upstream side to the joining segment is composed of 23 nucleotides. The 2 lengths are different, therefore this combination permits joining. For the lambda chain genes, the rule's the same, but it's in the reverse order. So for the heavy chain, the spacers for the variable and joining genes each have 23 nucleotides, so the number's the same, can't be joined. But the spacers that flank the D gene are 13 nucleotides in length, and so we don't have to break this spacer rule. The cleavage and rejoining of the DNA strands are thought to be carried out by endonucleases and ligases, respectively. Endonucleases cleaving and ligases joining. These enzymes recognize the heptamer and nonamer RSSs. The 2 genes that function in antibody gene recombination have been identified in mouse pre-B cells. These genes, RAG1 and RAG2, code for enzymes or other regulatory proteins. It's not totally clear. But these enzymatic processes are active only in the early stages of B and T lymphocyte maturation, when immunologic commitment is developing. So the gene products are not functional in mature cells, and thus the immunologic specificity of the B or T cell is maintained. So we've got all these genes mechanisms at work here. It's the chance recombination that creates a large amount of antibody diversity. If all events occurred randomly, somatic recombination in the DNA followed by RNA splicing could produce more than 1,000 varieties of the kappa light chains and a similar number of the lambda light chains. The random combining of heavy and light chains also greatly expands the number of possibilities. Imprecision in the joining of the V, D, and J genes also contributes to the diversity. This can be in the form of P nucleotide additions or exonuclease trimming. You can also have in-region additions producing changes in the specificity and reactivity of the antibody. In-regions are these very short peptides, often found near the third complementary determining region of the heavy chain. This region, the most variable region of the antibody molecule, contains amino acids encoded by the diversity gene region. Because the diversity segment is very small, the N region contains two joining boundaries where exons V and J join D at opposite ends. Also, immature lymphoid cells contain an M sign, which catalyzes the generation of N regions by the addition of nucleotides at the 3 end of the DNA strands. There's also extensive mutation involving variable region genes producing even greater diversity. Mutations can occur several ways. Point mutations, which are single nucleotide substitutions occurring in the variable region. Most mechanisms that generate diversity are active before any exposure to an antigen. Somatic mutation theory suggests that a small number of genes diversify by point mutation. These mutations occur after the cells have encountered antigen, especially during intensive immunization. The nucleotide substitutions are found only in variable domains of either heavy or light chains, predominantly in the CDRs. And finally, the antibodies produced by mutations in variable domain genes may confer a selective advantage to lymphocyte if they possess a higher affinity for the antigen. So cells coated with high affinity antibody are better able to interact with antigen and inducing continued the immune response. Characteristic of prolonged immunization is the increase in antibody affinity for the antigen as the duration of the exposure to that antigen increases, because the longer time increases the chance that a good mutation will occur. So mutations, chance recombinations, imprecise joining, and the addition of these N regions, this all contributes to antibody diversity.